Hey guys, it is Mr. Dave with the RayKitty Creation Workshop, and you are joining us for the RayKitty Science Project. So it is great to see you again, and we are going to be working on doing some DNA extraction, not distraction. I guess that was me, but DNA extraction, meaning we are going to remove and separate some DNA. Now, when we are doing this one today, before we kind of go into our materials and things like that, when we are doing this one today, our DNA that we're extracting, we're not going to end up being able to get down to a microscopic level to see those little double helixes in our DNA, but I do have kind of an example of that we'll do also. So what we are going to be extracting the DNA from is strawberries, and we're going to try grapes, okay? So I'm going back and forth on whether I try the grapes. We'll see, but because I know that the strawberries work very well, and I'm going to use the grapes kind of maybe as a contrast and see how they do. But the grapes, they're kind of difficult to do, so I'm going to kind of show you why we're using strawberries instead of grapes. One of the reasons is that these strawberries here have got a certain type of DNA structure to where they've got the strands. There's 8 of those DNA strands in there, and it makes them kind of nice and easy to mush and nice and easy to kind of turn into what we're going to extract the DNA from. So I'm going to show you kind of a comparison with grapes and why strawberries are the best. But I want to show you that comparison because if you're doing science at home, this is a very easy one to get into some high science if you would like to, or just some fun, mushy science. If you are doing it at home, it's always best to have something that you can compare and contrast. So this is a fun way to kind of check out the differences between those. So here's what you're going to need if you want to extract DNA from a strawberry. Okay, you'll probably need strawberries. So you'll have to get some strawberries. These are just store-bought strawberries. You don't need anything fancy, just regular old strawberries. You're going to need salt, okay? Obviously nothing fancy because this is plain salt. Yeah, so that's as unfancy as you can get. This is plain salt, so you're going to need some salt. This in these fancy little beakers here is dish soap. You're just going to need some dish soap, okay? And I have some toothpicks that I'll be using to mix that up with. You're going to need some plastic resealable bags. I call these baggies. So when I'm referring to it, I will probably say baggie. Yeah, I don't know where that came from. I've said it ever since I was a kid. So you're going to need a baggie. You're going to need some rubbing alcohol, okay? Isopropyl rubbing alcohol. This is 91%. Really, you're not going to notice much of a difference if you buy the 70%. Buy whatever's cheapest at the store, okay? So you'll need some rubbing alcohol. You will also need some coffee filters, okay? The coffee filters are definitely the easiest way. If not, you can use things like cheesecloth and all those strange kind of things, but coffee filters, they're pretty cheap, pretty easy to do. So you can use coffee filters. You're going to need some water, some cups, and just, general spoons, things like that. That's what you're going to need for your DNA extraction. So what we're going to do, another thing that I have here that we can kind of talk about DNA is this crepe paper, okay, or the party streamers. Okay, we have these party streamers. This is a fun way of kind of explaining the DNA and showing a little bit about the DNA, especially to your younger crowd or to the older students if you want to get into the actual cell structure, the structure within the DNA. And so the DNA is part of the cell, so not the cell structure of the DNA, but the actual structure of the DNA. Now, a fun thing to do with that, I'll move this, see if you can see this here, and a way to kind of use that as an example is if you take and draw a couple lines. Now, the reason we're using this particular kind of party paper stuff is if I draw a line on here, don't make fun of my artwork. This is a wonderful line. That's supposed to be a nice, beautiful line. It's not a nice, beautiful line. I'll try and go this way, see if I can make a nice, beautiful line. That's a terrible line. So these lines here that I'm making, okay, and then I'm going to draw just like to make a little ladder that goes up. You're going to draw just a little bitty ladder. Okay. Now, if you're doing it with some of your higher level students, you can get into the whole T, C, D, G, and all those kind of things that are going to be in this structure. But for now, we're just going to do this just for sake of time. As I said, if you want to do T is always bound with the A, and the C and the G are always bound, you can get into that with a little bit higher level students. But for sake of time, I'm just going to draw this little ladder. Now, when we say a double helix, the reason this stuff is fun is you can see through it on both sides once you've drawn on it. So we can see that we have that little helix structure there. Those lines would represent the little lines that are going to hold all of our little tiers together in our DNA strand. So this can be a good way to represent a double helix. The double helix means this little line and this little line are going to wrap around each other and give us that nice little twist there. So we have a nice little DNA strand example that you can use there. As I said, this is a good one, even if you are with the older students, to really get into the DNA structure of one particular thing, and then you can get into, hey, this DNA has this particular structure, that kind of thing. You can get pretty in-depth with a piece of party streamer and a marker. Okay, so this is a fun one, and you can, like I say, get pretty big on the science with that. But I did that so I can show you that our DNA that we're going to get to, we are not going to get down to seeing anything that looks like this. Okay, this is something that most normal microscopes that you see wouldn't even be able to see. Okay, it requires special microscopes to see that DNA. So we're not going to get down to where we see that, but we are going to separate and extract the DNA from our strawberries. So we are going to extract that DNA, and it is the same method that they use, just a very simplified version. And it is that same method. Extraction's about the same no matter what you're doing, whether it is on a cheek swab that you send into whatever to see if my grandparents were really my grandparents. I think they probably were. But yeah, if it's something like that you're doing, it's the same process. They just have it kind of refined, okay, instead of mushing things in a baggie, okay? So we're going to go ahead and we are going to, I'm going to keep this out so we can kind of use that as an example if we end up needing it. So I'm going to put this right over here. This was my little one that I was drawing to make sure everything would work. I'm going to put this over here too. And I'm going to move my crepe paper. We're going to get to that DNA extraction, okay? Now this is a very easy process. It is... I was gonna say foolproof, but wait till you try and do something on TV. You'll find out if something's foolproof. Yeah, you try it on TV and you're like, I have never had this fail before. And then you try it and you're like, I just failed so hard. So, but this one's pretty hard to mess up. The stoichiometry, okay, remember stoichiometry is our ratios, is not super important in this process, okay? You wanna get things pretty close, but it really isn't very, very picky about everything. So I've got some beautiful strawberries here. They smell wonderful. I'm going to pick this strawberry and I'm going to pick this strawberry. I picked these ones because they're pretty big. This takes about 2 strawberries, okay? You can use more, you don't have to, but this takes about 2 strawberries. If you're doing this with multiple students, teachers, 2 strawberries is a pretty good count. If you're doing this at home, parents, you can get more of them and make more mush, okay? So it really depends on how much more mush that you want to make, but you can get away with 2 strawberries pretty safely. So I have my 2 strawberries and my plastic bag or baggie. As I said before, we are going to try with the grapes. Now, grapes have got a different DNA structure, and it makes some of the portions of this a little trickier, okay? So I'm going to get a couple grapes in here. Now, as we can see, our grapes are much smaller than our strawberries. And so I am going to get, what do you think, maybe 3? No, I'm going to do 4. I'm going to do 4 grapes. So I've got 4, I'm going to do 5. I'm going to do 5 grapes. So it looks like about the same. What do you think? There you go. I like how I asked you. That was my Dora the Explorer, yeah, kicking in there. How many grapes? Yeah, so we've got 5 there. I'm gonna stick with 5. And I'm going to put these ones back over to the side right over here. So we have our grapes, we have our strawberries. We're going to put each of them in a bag. Now, with your strawberries, something that you're going to want to do is take off this little green top part. Okay, take off the greenery. We're not going to need that, and it's just going to kind of get in the way when we're mushing it. Okay, mushing is a scientific term, meaning it's not a real word. Okay, so you are going to mush these or moosh these. If you've ever been in my classes, I will say moosh. So you are going to moosh these. That is a scientific term. So we've got our strawberries. There you go. You will notice I put a little paper towel down. That's just so we're not making a big mess all over the table there. And it gives me something to wipe my hennies on. So we're also going to put the grapes in here, and we're going to notice a difference pretty much right away when we're working with these two different materials. So we have our grapes and we have our strawberries, okay? The strawberries, what you're going to want to do is mash, mush, smoosh, however you want to do it. With these here, you're going to mash them. So you're going to mash them with your hands. Now, if you are working with students, you will want to point out, don't use your fingernails, because if you use your fingernails on the baggie, you're going to tear a hole in the baggie. You're going to end up shooting strawberry across the room. So don't use your fingernails. We're just going to use the pads of our fingers. You can put it down on the table and moosh it on the table, but you want to mush this up. Now, this is, like I was saying, a thing that kind of makes this a fun one even for the little bitty guys because they get to moosh things. So if you are doing this as kind of a group project, if you're doing it at home with several different family members, you could have the little guys do the mooshing and the older guys do the science part. Or if you're doing it in a classroom, everybody can moosh. But as we can see, these strawberries, they mush pretty easy. Okay, we have a pretty easy time smooshing or mooshing these strawberries. Now, the better you smash them, the better they're going to do, okay? Because when we're smashing them, we're actually breaking down those little cell walls, okay? Remember, the DNA is part of the cells. So when we're smashing them, we're breaking down those cell walls. And so this is actually a very scientific step, even though it looks like you're just mooshing. Okay, so I'm mushing these guys here. The better we mush them, the better we'll get. I've got a pretty good mush right there. Okay, so I'm going to just kind of, you don't have to make the sound, it's completely optional, but it works a lot better if you do make the sound, the sound. Okay, so I've got some mushed strawberries. So we're gonna try that also with the grapes, and we're gonna see what the first difference is. Now with the grapes, when I'm squishing the grapes, okay, I am getting some of that juice out of there. That's how you get all that grape juice and those wonderful things. But we're seeing that they don't break down quite the way that those strawberries do. This is one of the differences and one of the things that's going to give us a harder time why I'm saying, hey, let's use strawberries instead of grapes. One of the reasons is they're a little bit harder to mush. Okay, we can get some of that juice out of there, but we have a lot of the portions here that are not going to render or not going to smash quite as well as those strawberries. So my grapes gave me a little bit in there for kind of a comparison and contrast. You can see the difference here, how our grapes are still nice and clumpy, right? Whereas our strawberries are nice and broke down already. Okay, so we can see that our strawberries are broke down and our grapes are still pretty lumpy. Okay, so this is one of the things that makes the grapes just a little bit more difficult. Okay, so our next step for this, now that you've got your material nice and mooshed, is to, we're going to make our separating solution. Okay. Our separating solution is when we're going to combine some of these materials over here. So you're going to want to do it in a cup. Okay, I've got a clear cup here so you can kind of see what's up. We're going to use our detergent and we are going to use our salt and we are going to use some water. Okay, so I'm going to pour out about half a cup of water. Here I've got, let's see, I'll go down to about half cup. That looks about right. As I said, this one here, the stoichiometry is really not super important. You want to be very close, but it is enough that you can allow the students to work the materials to take the measurements, and you don't have to like brush off and get exactly right, but you want to get pretty close. So I've got half a cup of this, okay, half a cup of the water, and I'm going to put that in my cup. There you go. And we are going to get some of our dish soap. Now, as I said with that stoichiometry, these little plastic spoons are not exactly 1 teaspoon, okay? They are just a spoon, okay? So we're going to use 1 spoon, and it's okay if it's not exactly a teaspoon. If you have a teaspoon, if you are teaching more of a chemistry end, or you want to really stick with the stoichiometry, this is 1 teaspoon, but I want to use this as an example that you really don't have to be dead on with that. So I'm going to get 1 teaspoon of my dish soap. Now, if you remember, in one of our previous episodes, we talked about polarity, right? And we talked about dish soap being an emulsifier, okay? That's exactly what we're going to do here, is we are going to emulsify or break down some of these cells in here, okay? So I've got that. We're going to get some of our salt, and I'm going to get a different spoon. That way I don't get the salt all stuck to my spoon there. And we are going to get 1 teaspoon of salt, okay? And you notice I kind of mixed in my dish soap a little bit already. So I'm going to actually, now that it's had a chance to set, now that it's mixed in a little bit, I am going to put 1 more teaspoon of our dish soap. Okay, so I put 1 teaspoon of dish soap, 1 teaspoon of salt, and then I'm going back with our dish soap. You don't have to do it that way, but it just seems to make it a little bit easier for stirring. Okay, so it's not a... if you end up putting 2 teaspoons of this in first or that in first, it doesn't really matter. It's just when you've done this with a lot of students, a lot of times you try and find things, little tips and tricks that make things a little bit easier. So that one will make this one just a little bit easier, and we are going to stir this up here in this cup. Okay, so now that I've got it stirred in the cup, you can see it's pretty well broke down. We have this nice cloudy little mess right here of our separating solution. So this is our separating solution, okay? So I'm going to put my spoon over here. I'm actually going to put my spoon on my paper towel so that I don't end up getting it on my rag there. So we've got our separating solution, and we have our material here that we had made when we were smooshing everything, okay? So what we're going to do now is I am going to pour this. I'm going to take these grapes, and I'm probably just going to set them to the side. I don't think we're even going to end up working with those because this is why we did that compare and contrast. When we look, I've got some pretty solid material there. I'm going to have a hard time getting this to this point, okay? Now, another reason that our grapes, even if you were to take it a little further, the way that the DNA is aligned within the grape, you really are not going to get much, okay? So it's fun to do a compare and contrast. If you want to take your grapes all the way through and make a solution for your grapes and go all the way through, teachers or parents, you're not going to get a lot of DNA out of them. So that's what I was going to use as an example, but we can see that this really isn't going to work. So I appreciate the compare and contrast, but we can see I'm going to just step away from these guys. So I'm going to put the grapes over here, and I'm going to move my solution into here. Now, this is one of the points where it is very important that you did not poke little bitty holes in the bag. If you have a student, especially the younger ones, that may end up poking them for this next part, because we are going to pour this right into our bag, you may want to go ahead and put this bag inside of another bag, okay? So there's nothing wrong with that. That'll make it a little bit easier for them to not poke a hole in our bag. But what we're going to do is this is our separating solution, and we are going to pour this into our strawberry mush, okay? Now, you'll notice I'm remixing it because I chatted for a little bit there, and so some of it settled to the bottom. But this is that emulsifier, just like we talked about with our polarity. This is the emulsifier. So I'm going to pour this solution into here, okay? There you go. Now we have a very gross little mixture here. One thing you are going to want to point out is they're going to mix this. You're going to mix this in the bag. I'm getting as much of the air out of the bag as I can. because you don't want to have like a really pressurized bag. Have a kiddo squeeze it, and then you have strawberry all over the place. Just because you have soap in there does not mean this will be easy to clean up. So I'm getting as much air out of there as I can. Another thing you will want to try to avoid, that dish soap has a tendency to bubble up. So as they're mixing this, I would tell them do it pretty carefully because they don't want it to get too bubbly. Okay, so you're going to try and avoid those bubbles as much as you can. Okay, so I'm mixing this up. I'm mixing my strawberries in there. Okay, and what's actually happening here is it is actually dissolving that cell membrane. Okay, the emulsifying, like we were talking breaking down, is actually dissolving that cell membrane. So this is doing the same thing it does. The reason it makes it easy to wash your stuff off when you have soap is that this is breaking down the cell membrane in our strawberries. Okay, so this strawberry little goo here, as you can see, I'm kind of mixing it while I talk. I'll get to where you can kind of really see that because it's pretty nasty. So this stuff here, those cell membranes are breaking down, and so what that does is that kind of releases the DNA, right? Because we talked about that DNA is a part of the cell structure. So we've got this. It should be pretty mixed up now. You can't over mix it, but at the same time, you don't want to get it too bubbly, okay, too frothy. All right, so we've got this in here, and what we're going to do now is I'm not going to get my original cup. I'm going to move over and I'm going to get a new cup, okay? So I've got this cup here, and now we are going to separate our liquid kind of from some of those solids. We're going to get those solids out of there because if it is still solid, it hasn't broke down. Yeah, so the part that is liquid is now the broken down cell membranes along with our dish soap and our salt and things like that. So this is the part I'm going to make sure this is sealed before I set it down. Yes, I say that from experience. I have set these down and then I just had a lap full of strawberry dust. So, not dust, but goo. So I'm going to get one of my little coffee filters here. Okay. Now this part here, again, may get a little messy as far as the next part, just messy on your hands a little bit. If you are careful, you won't be making a mess on the table or anything. But what you can see is I took my coffee filter and I put it to where I have a nice little cone for my material to go into, but I also wrapped it around the outside. That is just because I don't want my coffee filter dumping right into my material. Okay, so I want my coffee filter to stay in place. I'm going to actually hold the cup. I'm going to hold the coffee filter or attempt to as I'm pouring it. Now it may take two hands to pour from my little baggie here, but I'm going to try to hold this and pour that. There you go. All right. And then I'll be like, and for my next trick. Yeah, because we'll see how this goes. So I'm going to pour this. And what we're doing now is we are just separating the solids from the liquid. Okay. Now we will still have dish soap in there. We will still have, let's see, there you go. Yeah. So you can see how that is, the lumps are coming into the top of my cup here. And the rest is kind of washing down through that coffee filter. Okay, so this portion here, this part here takes a little bit to let that drip through there because we want that to go through our coffee filter. So I'm going to keep pouring this in here while we're talking a little bit about DNA. Okay, so our DNA is the kind of the little sequence or the part that makes everything up and makes us different. Okay, different DNA will give us different results, right? Everyone has different DNA. We all have similarities in our DNA, and that's the portion of our DNA that makes us. If you align DNA a certain way, then you end up with a person. If you align DNA a certain way, you end up with a strawberry. Okay, so we have different ways that those DNA will sequence that end up giving us different results. Certain DNA will give us green eyes, blue eyes, those kind of things. And so that's how we can kind of tell, or if you watch like shows with a lot of like detective work and things like that, they'll say, oh, well, this person has that DNA. And that is because our DNA is unique to each of us. We all have a very special DNA, and our DNA is only our DNA. Okay? So I'm going to let that kind of soak through there for just a second while I'm getting my other stuff ready. I am making sure that I reseal this guy up real good before I put it in here. Now, you'll notice I didn't use all of that. You don't have to use all of that. If you have multiple kiddos or students you're working with, you can separate that in between multiple students and you'll be fine. So you just need to make sure that you've got some of that juice and water there. Okay, so we've got this in here. What we have is our separating solution and our strawberry DNA. Okay, so I'm going to take this. I'm going to put this over here so I can put the rest in it. Actually, I'm going to get a different cup so I don't make a mess all over my baggie. So I'm going to get this here. I dumped all my lumps in there. Here, I can filter that right back through real quick. Just a sec. So what we have here is now our other strawberries are back in there with it. So I'm going to do it in this one here. And I'm going to give it a squeeze this time and get that juice going a little bit quicker so I can get it through there. Because I let go of my coffee filter. There we go. This was that. Remember how I told you, yeah, this is real easy unless you're trying it on TV? All right, so I'm going to kind of speed up the process a little bit. I was trying to avoid doing this because it gets your handies messy, but we got plenty. So that was twice. One more try. Here. Okay, I'm going to try this one more time. Here we go. Get my strawberries in here. Get my coffee filter in here. I'm going to get a little bit more coffee filter around the edge this time. I'm going to pour this in here. There you go. And now, here you go. Third time is a charm. I'm going to wrap up the end of this and give it a squeeze. There we go. Nice. So this time I don't have any of those little strawberry parts in there. Okay. So now we've got our strawberry. This is the first time I think I've ever milked a strawberry. Yeah, there you go. So that is not science. Yeah, don't take that one. That is not science. You don't milk strawberries. So there you go. But strawberries and milk though. All right, now we have some of our juice here. What we're going to do next is we are going to get our rubbing alcohol. Okay. And we are going to, when you do this, you want to be gentle about it because you don't want to stir up a bunch of bubbles there. So we're going to take our alcohol and just gently pour it down the side. You want about as much alcohol as that other fluid that you had. Okay? So we've got our alcohol in here. And what you can see is this is called precipitation. So we are precipitating the DNA, which is just like when the rain separates from the clouds, we are precipitating or separating that DNA. Okay? So now our DNA is separating and it is coming to the top. And you can see that little line there, that delineation where we have our goo that we had, and that DNA starts to separate up to the top. So this is called precipitation of the DNA. And so those DNA strands start to form on the top of our cup. Okay? Now these strands, I've got my toothpicks here, and I'll see if I can capture one of those little strands on my toothpick. Now, if you look in here, I'll be pretty gentle with this. If you can kind of see on there, I'm going to lean this cup very gently, and we'll see. Now, the strands, oh, I got one. Look at that. There you go. The strands look like snot, right? Ew. But what we actually have here is we have separated the DNA from our strawberries, and then we've precipitated it. The DNA will separate up, and those little portions there, if you leave this set for about 5-10 minutes, you will have all kinds of those DNA strands all along the top, and you can get a lot into the science behind the DNA, right? So what we did was we extracted the DNA from a strawberry, right? So this is a fun thing. You can take it up to that next level. That little snot there, if you do have, is not really snot, it's a DNA. But that group of DNA, If you have a microscope, you can kind of check that out. You won't be able to get to that helix, but you can kind of see how those cell walls have broken down. So this is extracting the DNA from a strawberry. I'm Mr. Dave, and as we say when we end everything, science shows you matter.